Hello, my dear students. It's been a while since we've had a lesson. I know, I'm a very bad teacher and I probably don't deserve to call you my students. I hope that you will forgive me. Truth is that during this time I didn't make any movies because I couldn't find anything worthy to speak about. I know that most of you expected from me to make another episode of my famous Planet X Doomsday Fearporn. But sadly, despite going through gigabytes of data, I wasn't able to find anything of actual importance for the things which I've stated already in my movies. And I really don't like to speak about things which I've discussed before. So I will have to disappoint you once again. I still didn't manage to find the planet X or whatever might be the thing up there that seems to disturb the solar system and the interplanetary magnetic field. Sorry, but I tried. Luckily, we're living in a very special times when the difference between most of conspiracy theories and scientific facts is around 6 months or so. I would however argue that in fact it all depends on the level of apparent craziness in each particular conspiracy theory. The more crazy it sounds to some average Joe, the longer it will take for it to become a generally approved fact. And since all claims about the planet X were generally considered as pretty much totally crazy, around 9.2 in 10 on the craziness scale, at the time when I started to record my movies, 2013, it took couple years for the normal part of our society to slowly but steady succumb to my madness and admit that there might be actually some big unknown celestial body at the edge of the solar system. They just called it Planet 9 to not look stupid in the eyes of conspiracy theories. And now, at the end of year 2029, we're at the point where I can leave the search of Planet X to actual physicists and astronomers. And honestly, considering my very limited capabilities and resources, they probably have much bigger chances of success. Still, I wouldn't be myself if I wouldn't try to use their calculations and see if I can help them just a bit. Actually, I did so already and found couple interesting findings, which might or might be not candidates to be recognized as the actual Planet X. However, I will show you those findings at the end of this movie, as right now I want to speak about a completely different conspiracy theory, which is in fact so low at the craziness scale that it turns into a generally accepted fact just as we speak and right before our eyes. And I think that many of you will agree that there is no better example of a conspiracy theory turning out to be true than the outbreak of a virus called SARS-CoV-2. As most of you know, in late 2019, a new type of virus emerged in the Chinese city of Wuhan. First cases of an unknown pulmonary disease were noticed in a local wet market, which happens to be located couple blocks away from the Wuhan Institute of Virology, which specializes mostly in researching bad coronaviruses. Of course, couple days later, we learned that this new and unknown disease is being caused by a novel coronavirus that most likely originated from a bat. What a surprise! And then some people started to wonder where could this virus originated from? Obviously, a complete mystery. Clearly, we face a truly impossible task of connecting two dots and finding the causality in A pointing to B. But obviously, we, the common people, aren't the ones allowed to connect the dots by ourselves. Clearly, we don't have necessary credentials to make it properly, and we should leave it to people with actual authority in this field, as only they can do it properly. Simple commoners can only listen to them and accept their words of inborn wisdom without question. And according to people with authority, this virus started to spread like a wildfire because someone ate the meat of infected bat which he bought on the wet market. Of course that bat meat was being sold there. There is no need to prove it. Accept and obey. It could be also a pangolin. Or even better, pangolin infected by a sick bat which just happened to visit Wuhan. And the sad part of this story is that such explanation was adapted by mainstream media as the generally accepted narrative and used to discredit anyone with more than two brain cells and the ability of using them to think by himself and making the most obvious and logical conclusions, even if they are against the claims of people who consider themselves superior to the stupid human flock. And then, slowly but steady, more and more evidences started to emerge from the information chaos, and it became more and more obvious that just like in most of other cases, the most logical and the most obvious conclusions are the ones that are correct. As for today, 2 years after the virus outbreak in Wuhan, even the most prominent brands of mainstream media agree that lab leak seems to be much more possible than a bat infecting by the pangolin. But this shamelessly obvious conspiration doesn't end here. In fact, from that point it seems to steadily progress into territories on some lesser known Tom Clancy's novel, or even more action packed but less believable stories written by Dan Brown. Only this case, author wasn't too creative, since the most critical plot twist happens when we learn that the greatest health and economic crisis of the 21st century was in fact funded by legal, registered, and law abiding US taxpayers, only without any kind of their legal consent. So it makes some sense that when it comes to global pandemic, no one should have better knowledge than the guy who provided the funds needed to develop and research the virus responsible for the ongoing crisis. If there is one guy who should tell us something useful about the outbreak of a bad coronavirus, then it has to be our beloved Dr. Mengele Fauci, the greatest medical authority according to mainstream media and the approved narrative. Thing is that it would indeed make sense if Dr. Fauci would be capable of making any correct prediction or factually valid statement. Sadly, it seems that providing dollars is the only thing about gain of function research that Fauci is actually good at. Worse when it comes to practical knowledge and understanding of novel coronavirus, as most of his predictions look somewhat like meteorology based on fortune cookie telling. Normally, it should be possible to tell who's the evil mastermind in this James Bond like story by pointing out a guy who thinks that cruel and prolonged tortures of beagle puppies are fully justified if we call them scientific experiments. I mean, what kind of a sick psycho can approve any form of such activity? Yeah, well, according to the mainstream narrative, such way of thinking is in fact based on totally unfounded stereotypes, and as such, it should be completely rejected in a preferred social model. The fact that someone likes to spend time by inducing agonizing pain to innocent puppies has absolutely no real impact on him being a truly beloved national hero. But just in case of some non-compliant leftoid who for some reason can't see the greater good that is being achieved thanks to those scientific experiments, dozens of fact-checking sites will eventually help to overcome the undesirable bias, allowing him to successfully ignore the voice of naturally inbred human conscience and accept Fauci as the true modern-day messiah that he obviously is, according to the mainstream media. And generally, I could end this story right now and move forward with this lesson, but I wouldn't be myself if I wouldn't try to spice things slightly up with a solid dose of a conspirational controversy. You see, all good conspiracies have the tendency to reach much deeper and spread much further than we might generally suspect. And this particular conspiracy isn't probably different in this regard. However, this is where I have to enter the twilight zone, separating unverified speculation from generally settled truths. In shortcut, subjects presented in this movie are more than enough for me to become a very prominent public enemy for the YouTube platform, what might possibly become the reason for my channel to earn a permanent and definitive ban. It doesn't mean, of course, that I won't make everything to prevent the impending ban from happening by avoiding everything what might be labeled by the YouTube censorship committee as the widely understood disinformation. This is exactly why I will try to not make even a single claim that goes beyond the generally accepted state of affairs. So to be perfectly clear, I don't claim that Bill and Melinda Gates Foundation has anything to do with the gain of faction research and or the Wuhan Institute of Virology. All I do here is to present you some random points of unverified possibility, and it's up to you as a viewer to draw or not to draw the lines between those points. In this particular case, dear students, it's for you to decide if it's just a pure coincidence that the virtual simulation took place just a month and a half before the real-life scenario, or if there is some hidden motives behind everything what is taking place since the outbreak in Wuhan. I don't consider myself reliable enough to tell you what is or isn't true. I can, however, try to make everything to ensure you that the information presented here is fully consistent with the generally accepted science, and then each one of you will be able to verify it with an independent opinion of someone with proper knowledge of presented subject. But although due to my efforts of maintaining scientific consistency, I won't speak here about satanic rituals with reptilians drunken with virgin blood feasting on meat of newborn babies, but still I can promise you a solid dose of drama that goes rather grim in the progress of this story. Of course, I'm not so naive to assume that being factually or scientifically consistent is enough for the YouTube team to leave this movie and or my channel just as it is. This is why, just in case, I will make a complete backup of my channel, so that those 7 years of my career won't be completely lost. They won't get me so easy. Guerrilla warfare is my national specialty. Okay, that should be it as for the introduction. Now it's time to deal with some actual science. Don't be scared. As always, I'll try to make this edible to an average commoner without specialized academic education and or genius level of intellect. And if you manage to actually understand some of my lessons about plasma physics or relativity, it should be for you a piece of cake. But first of all, I need to speak about couple experimentally proven scientific facts that can work for me as a foundation for everything else. As I said in the beginning of this movie, we've entered a rather strange period of history when a conspiracy theory turns into proven fact almost overnight. However, a conspiracy is not equal to any other one. Planet X doesn't prove flat Earth. As strange as it might sound, for a guy who spent last years researching Planet X while trying to discredit Einstein, I actually trust in science, especially in the one that is backed up by direct evidences. And in case of the conspiracy that seems to surround the ongoing pandemic, experimental science is pretty straightforward in deciding which part of the conspiracy can be verified as being true and which exists only within the boundaries of a cavity sealed by a double-layered tinfoil hat. I'm pretty sure that some of you won't like what I have to say, but direct evidences prove without any real doubt that the virus known as SARS-CoV-2 is a real physical entity. Sadly, the only way for you to make me change my mind is to show me even one, preferably peer-reviewed, article presenting the idea that people all over the world are getting sick due to chemtrails full of more galleons that are activated by 5G frequencies. I might be a bit too harsh, but I've seen quite a lot of people who claim that SARS-CoV-2 was never observed, isolated, or sequenced. So let's make it clear once and for all. Either they never tried to fact check their own claims, or they are knowingly spreading lies. Truth is that even a basic Google search is enough to learn that the virus was actually isolated and sequenced by hundreds of scientists all around the globe couple weeks after the outbreak. But there is of course much more than that. Although viruses are known to be much smaller than any kind of multi or unicellular life form that is known to us, it's still possible for a virus to be directly observed under an electron microscope. And there is actually quite a lot of such microscopes, with some of them being available for the public to use. Since it's actually possible to use such technology to see an actual atom, observing a virus isn't in fact a big deal. In fact, probably there is no other microorganism on this planet that gets as much attention as the coronavirus SARS-CoV-2, and the amount of data that is available to anyone is pretty staggering even for me. And of course, there is also at least just as much data regarding the illness that is caused by that virus and is known as COVID-19. And since you've reached such remote point in the movie, even with my completely defective guidance, there won't be a better time for me to disappoint some of you the second time in a row by stating that in many cases COVID-19 is much more severe than the seasonal flu and that among certain age groups it has significantly higher rate of mortality. I am terribly sorry, but I promise sticking only to hard scientific data, And in this case, such kind of data gives me a rather clear and self-consistent description of the illness known as COVID-19. Although data regarding things like infection to death ratio can be skewed or manipulated in order to present the facts in a favorable way, numbers representing the all-cause death rate among different countries don't leave any place for speculation. In most European countries, every legal citizen has his own personal ID number given to him soon after birth, so it would be very hard for anyone to fake those statistics. And while there might be some understandable doubts regarding the causes behind people dying in greater than normal numbers in 2020 and 2021, the increase of death rate can be noticed on most of those charts, coinciding nicely with the outbreak of SARS-CoV-2. However, I can bet that after 2 years since the outbreak, most of you had already some kind of a direct or indirect experience with the SARS-CoV-2 virus, and there is no need for me to prove you that it is pretty much real and it might often be much more severe than a flu. But I think that this will be a nice starting point in my efforts to make some things clear for all of you. So now let's take a closer look at this particular specimen of a coronavirus and see what it is made of. As I said a couple moments ago, SARS-CoV-2 is probably the most known virus on this planet. Sequencing its genome was the first thing that was done just after its identification and isolation. Now, 2 years after the outbreak, you can use online tools that utilize data gathered with electron microscopes to visualize all of its proteins in 3D, or even simulate their behavior. In shortcut, if you're a professional virologist, you can get for free a lot of high-end toys to play with. Even for me, it's more than enough. Just look at this site. To be completely honest, the only way in which I can use such powerful research tool is to have some cool looking animation in the background as I verbally present the most important information about the virus and its structural composition. Viruses, together with bacteria and fungi, are the main three causes of all known infectious diseases. What makes here the most important difference is that viruses are tiny compared to any known unicellular organism and are very often much more infectious. What makes viruses especially dangerous is their ability to break into healthy cells of infected organisms and hack into the genetic code, where they can execute scripts of replication, due to which the virus is multiplied into massive numbers of copies, which can then infect more cells in the hosting body and or allow the transmission to a new and uninfected organism. Compared to that, diseases caused by bacterial and fungi infections tend to be more localized in specific parts of body and are mostly much less contagious, with couple exemptions. All viruses are generally assemblies of couple different proteins, which form their structures, characteristic to specific families of viruses. Luckily for us, we will deal only with the coronavirus family, as the ongoing pandemic is being caused by a virus that belongs to it, just as couple other known viruses, with SARS-CoV-1 and MERS-CoV among them. SARS-CoV-2 is made of 29 proteins, with 4 types of them being structural proteins that make the virus to look just as it looks, and for now I will focus mainly on them. Nucleocapsid N protein is an important structural protein that packages viral RNA into helical ribonucleocapsid (RNP) and interacts with the other structural proteins during viron's assembly, leading to genome encapsidation. The N protein is produced in high abundance during infection and is highly immunogenic, thus a potential target for vaccine development. Membrane, or the M protein, is the most abundant structural protein and defines the shape of the viral envelope. It is also regarded as the central organizer of CoV assembly, interacting with all other major coronavirus structural proteins. Homotypic interactions between the M proteins are the major driving force behind viron's envelope formation, but alone is not sufficient to virion formation. The M protein induces a strong humoral response, thus it can be a potential immunogen in therapeutic applications. The envelope, or the E protein, is the smallest of the major structural proteins, but also the most enigmatic. During the replication cycle, E is abundantly expressed inside the infected cell, but only a small portion is incorporated into the viral envelope. The majority of the protein is localized at the site of intercellular trafficking, where it participates in CoV assembly and budding. Coronaviruses are named for the crown of protein spikes covering their outer membrane surface. Early work on the novel coronavirus has focused on those spikes proteins, also called S proteins, because they are the keys that the virus uses to enter host cells. In both SARS-CoV and SARS-CoV-2, the S protein binds to a receptor called ACE2 to hack its way into host cells. Out of all structural proteins, the spike is probably the most sophisticated one. First of all, we can define two separate stages, conformations, of the spike life cycle. First is the prefusion state, images A, B, and C, in which the spike remains until it won't bind to the ACE2 receptor on the membrane of a host cell. When this happens, it triggers couple processes that turn the spike into its post-fusion state, image D. For now, let's however look closer at the prefusion state of the spike, where we can divide it into two fragments, S2 in the bottom and S1 on the top of spike. S1 is composed of three proteins of the same kind, and each one of them contains a fragment called receptor binding domain, RBD, which works like the access key for the keyhole of ACE2 receptor. There are also two separate forms of the prefusion conformation, one that is called RBD up, where the binding domain is being exposed and ready to bind to the ACE2 receptor on the host cell membrane, and second called RBD down, where the spike is closed and the binding domains remain hidden. Binding to ACE2 enzyme is possible only for a spike in the RBD up state. Here's an animation which shows up and down forms of the S1 part of the spike in its prefusion conformation. After the binding, subunits of the spike's S1 fragment are being cut out by certain enzymes called TMPRSS2 in a process called cleavage that results in exposing the S2 fragment, which then undergoes significant structural changes that allow it to connect to the host cell membrane and initiate the entrance of virus into the cell. But since all of this is a quite complicated process, I will now show you a nice animation, which was brutally stolen by me from YouTube and which shows the entire process of ACE2 binding and the subsequent membrane fusion during the infection of SARS-CoV-2 coronavirus. Of course, after the fusion of membranes, genetic code stored in the viral envelope is being written into RNA of infected cell, initiating the replication of the virus. But this process is not the subject of today's lesson. What I want to discuss instead are the properties of SARS-CoV-2 spike protein and the processes involved in the interaction of spike with a host cell. There are a couple reasons for me to choose this particular topic. S protein has couple interesting capabilities that can't be observed in any other protein of SARS-CoV-2, and is being used as the primary target of immunization in all approved anti-COVID-19 vaccines that are currently available on the market. And this is what makes it a rather controversial subject of still ongoing scientific discussion. Let's begin from this article, which you can find for example here. I think that the title tells all. The paper was peer-reviewed and published in Journal of Biological Regulators and Homeostatic Agents last year, and it summarizes the most important information from other publications regarding the spike protein. Of course, I wouldn't be myself if I wouldn't verify the things that are stated here, and as you might guess, those things seem to be quite worrisome. First of all, at the time when I'm making this movie, it's a well-known and established scientific fact that spike proteins by themselves are the most dangerous ones of all structural proteins in SARS-CoV-2 and are capable to cause a severe damage to healthy cells without the rest of the virus. In this particular case, it has been proven that mice injected with the spike proteins suffered lung damage. But it's just the beginning. From now on, it will get only worse. Going further, it turns out that SARS-CoV-2 spike protein induces inflammation. And for those who don't get it, inflammation isn't a kind of immune system reaction that is desirable. As we can learn from the article, spike protein potently induces inflammatory cytokines and chemokines, which are the main cause of post-COVID-19 Kawasaki-like syndrome that is being observed in some number of children infected with the virus. It's worth to mention that any other structural protein of SARS-CoV-2, membrane envelope and nucleocapsid, doesn't cause such kind of response. Interesting, but let's go further. I recommend you to pause and read the articles that will be presented on the left side of the screen, as there is too much of them to discuss each one of them individually. In shortcut, inflammation caused by the spike protein is actually the least of problems. Much worse is a process called here as apoptosis through autophagy. For those who don't know what is the meaning of those terms, apoptosis is a kind of self-destruct algorithm of a cell, while autophagy is a process in which cells are being eaten up by our own immune system. I will, however, focus on this particular fragment. We can learn from it that in the moment when a spike binds to ACE2, a pro-apoptotic response of the host cell is being triggered, even before the virus enters that cell. Are you getting scared already? Then hold on a bit longer, as we're not even in half of the discussion. Another interesting property of the SARS-CoV-2 spike protein is its ability to deregulate and pass through the blood-brain barrier, causing inflammation of brain cells. And again, the amount of available information is so large that I'm not able to discuss it whole. Instead, I will show you again couple fragments for you to pause and read them. Take note that it's being clearly stated that even without the rest of the virus, spike proteins alone are enough to pass and damage the BBB. But before we go further, I want to point out three different mechanics in which the spike protein can potentially cause a serious damage to the brain of a healthy person. First one involves the spike protein inducing formation of microclots, what leads to infarction in the brain and causes acute ischemic strokes in people younger than 50 years old. Second mechanism involves the ability of SARS-CoV-2 spike protein to increase production of enzymes like MMP3, which is involved in traumatic brain injuries, or MMP12, that uses infected immune cells to enter the brain. Later authors of this publication speculate that this mechanism makes the infected immune cells to act like Trojan horses, helping the virus to infect the brain. The third mechanism which I want to speak about involves the reduction of ACE2 enzymes in the brain tissue, which is being caused by the binding of spike proteins. Reduction of ACE2 activity was recently reported in patients with dementia and Alzheimer's disease. Okay, that's it for now when it comes to the influence of spike proteins on brain. So now it's time to see what is the influence of SARS-CoV-2 spike protein on the DNA of infected person. Well, according to this study, in vitro experiments show that it is capable of significantly disrupting the DNA repair mechanism. According to the results, authors of the study found the abundance of spike proteins in the nucleus of an infected cell, next to a couple other non-structural viral proteins. And according to those graphs, the spike protein induced the strongest inhibition of DNA repair. Okay, but I've saved the best part for the end of the discussion about SARS-CoV-2 spike protein. Although the title of this article doesn't look too alarming, It's the interior what makes it quite dramatical. It's also worth to mention that it was published in a journal called Vaccines, so it's quite possible that people who are responsible for the production and distribution of COVID-19 jabs had the opportunity to read it before their products became available for the public. While the title probably doesn't tell anything to 99% of commoners like me, the general idea presented in the paper is rather simple. In shortcut, it's about ACE2 not being a receptor in a non-infected body, but being a type 1 membrane protein that is responsible for multiple physiological processes, with regulating blood pressure being one of them. The issue is that due to the influence of SARS-CoV-2 spike protein, ACE2 stops to work as a regulator and becomes a receptor for the spike. As you might guess, it leads to multiple unwanted effects, like the autophagy and subsequent apoptosis of affected cells, which I mentioned earlier. But the question is, how all of this affects the actual health of infected person? I really don't want to unnecessarily scare the vaccinated viewers, but according to this publication, just by binding to ACE2, SARS-CoV-2 spike proteins can lead to a very serious and chronic illness called PAH, pulmonary hypertension, which according to authors doesn't have a cure, can affect people at any age, and in most cases leads to death in around 2 to 3 years if it's untreated. However, even if treated, it gives the patient only 60 to 70% chance of surviving more than 3 years. What makes the PAH especially scary is the fact that it requires a sophisticated examination, invasive right heart catheterization, to be diagnosed, while its symptoms, shortness of breath, fatigue, or dizziness, are similar to other non-life-threatening conditions. Well, problem is that shortness of breath, fatigue, or dizziness are pretty common side effects of all currently available COVID-19 vaccines. And the idea of SARS-CoV-2 spike protein causing PAH is not just a theory but a scientific fact that was proven in laboratory. But this is still not all, as the authors suspect that the spikes binding to ACE2 might cause many other serious diseases and adverse pathological events in all parts of the body that include cells expressing ACE2. I'm pretty sure that many of you would now love to know how many of those who experience shortness of breath, fatigue, or dizziness after vaccination for COVID-19 will in fact develop PAH. Yes, I would love to know it as well, but I don't. Obviously, we all will learn the answer in around 2 to 3 years. Yeah, I know, it's not funny if you're vaccinated and experienced one or more of those symptoms. But what can I do? I just provide you the science, nothing more. But just to be perfectly clear, all those papers discuss the spike protein in its natural form, characteristic to the SARS-CoV-2 virus, and not the spikes utilized in approved COVID-19 vaccines. Of course, according to multiple fact-checking sites, spike proteins used in the available vaccines are not the same as spike proteins attached to the actual virus, and because they are somewhat different, they are also completely and absolutely safe to a healthy person. So there is no need to worry. Just take the goddamn jab. But of course, I wouldn't be myself if I wouldn't fact check the fact checkers. According to officially approved sources, there are three main differences between the spikes used in vaccines and those which are characteristic for the SARS-CoV-2 virus. 1. Spikes used in the vaccines aren't attached to the virus, so they don't cause COVID-19. It's true, however, in none of the presented papers, getting COVID-19 was necessary to experience the pathological events caused by the spike proteins themselves. Spikes used by some vaccines are genetically modified in order to remain anchored to the host cell membrane, so they are unable to circulate freely in the bloodstream of vaccinated person. It's true, but it doesn't prevent those spikes to disrupt the physiological function of ACE2 in all cells that are surrounding the infected cell, with spikes proteins being anchored to its membrane. Spikes used by some vaccines are genetically modified to remain locked in the prefusion state, what supposedly makes them incapable of binding to ACE2 and initiating the apoptosis of healthy cells. I'm really sorry to be the one who has to say it, but in this case, everything except the part about spike proteins being artificially locked in the prefusion conformation is a complete scientific and medical disinformation. Here, let me quickly prove it. Here's a paper titled Safety and Immunogenicity of Recombinant Stabilized Prefusion SARS-CoV-2 Spike Protein Vaccine. Title is longer, in which it's being stated clearly that the S2P, a prefusion-locked modification of spike protein that is being used in some of approved vaccines, Pfizer for example, can bind to ACE2, just like a normal SARS-CoV-2 spike protein does, what makes it a potentially lethal cytotoxin. In shortcut, no matter what's the reason, information provided by this source that dares to consider itself a fact-checking site is in fact a scientifically invalid medical disinformation, and as such it shouldn't be given on the first page of Google search results. Sorry, but those who live by the sword die by the sword. And here is the actual scientifically valid fact. In order for the SARS-CoV-2 spike protein to be considered somewhat harmless to a healthy body, it would need to be a. Locked in the RBD down state of its pre-fusion conformation, b. Locked in the post-fusion conformation. Only then spikes wouldn't be able to disrupt the natural functionality of ACE2. It has been experimentally proven that the ability of binding to ACE2 is greatly reduced in a spike-like construct that is artificially locked in the closed RBD down conformation, what makes it much less infectious and less harmful compared to spike proteins used in currently available vaccines. Thing is that such modification of the spike is at the moment only a possible future target for some next-generation vaccines, and as for now, no one seems to care about doing anything about the ACE2 damaging technology used in currently available vaccines. Okay, this is where I will for now end today's lesson of virology. However, it's not even half of the subject which I want to discuss with you. If by any chance my channel won't be nuked out from the face of YouTube after I finish uploading this movie, next one will be probably even more controversial, as I intend to look closer at the actual safety and efficacy of the approved anti-COVID-19 vaccines. But as for today, I have only one advice for you all. Keep trust in science. Class temporarily dismissed.